Can you hear that? Right. What makes animals special is they are complex arrays of cells which communicate to form intricate patterns of gene expression that vary precisely from one cell to another throughout the animal, throughout embryogenesis, and they determine the embryo morphology. One of the big challenges is to understand gene regulation and ultimately the entire transcriptional network. For this, we need cellular resolution descriptions of the gene expression, the morphology, and the dynamics. We need to quantify the inputs, the amount of transcription factor in the proteins, and the outputs, the amount of different mRNAs produced for every cell in situ in the animal. And this is really a challenge in microwave sort of analysis. So imagine a spreadsheet, a table, an atlas that records the position and location and other physical properties of all the cells in the animal. The atlas would contain many features such as the expression of multiple genes on a per-cell basis throughout embryogenesis. Imagine the computational sort of analysis that you could do with this atlas. We have built such an atlas from one of the earliest stages of embryogenesis in Drosophila. At this early so-called blastoderm stage, the embryo is a hollow ball of 6,000 undifferentiated cells. To measure gene expression and morphology, we have used an image analysis approach where we take three-dimensional stacks of the embryo labeled for the total DNA for nuclei and the expression of two gene patterns, one of which we use as a registration guide. So these images are then converted into a flat text file that describes the XYZ coordinates of each nucleus along with the gene expression levels. So such data from thousands of embryos are then registered into a single model. Our current data set records expression of 6 consecutive 10-minute time points and has data from protein expression of 21 transcription factors and the mRNA patterns of 100 genes that the factors regulate. So the challenge in this registration is that we need to do it correctly on a cell-by-cell basis. This is necessary because to create accurate biological atlas, nuclei move through stage 5, shown in these heat maps here, even though there are no mitoses. And different embryos have different sizes and have different numbers of cells, as shown in this graph here. And the relative expression boundaries of patterns vary from one embryo to sort of another. Now, these need to be correctly registered into your atlas. So we have built such an atlas of the blastoderm. It's publicly available at our website. So we are currently using the atlas to build computational models that predict mRNA expression patterns of genes in 3 dimensions from in vivo binding and chip data and protein expression patterns of transcription factors. We use a generalized linear model that takes 95 cis-regulatory modules to predict the output patterns of transcription per cell, shown on the right here for 2 CRNs. The modeling is ongoing, but as you can see, compared to the measured patterns on the left here, in some cases we're doing quite well, and in some cases we need more work. The bottom line is that gene expression patterns, the inputs and outputs of the transcription network, are extraordinarily complex and precisely varying. Okay. That's published work. This is what we're doing now. Creating an atlas of the rest of embryogenesis is challenging. Over the course of the next 10 hours and 3 major mitoses, the 6,000-cell blastoderm becomes a 40,000-cell animal with 70 different tissue types. One of the challenges here is the density and complexity of cells, which make the embryos opaque. We are very interested in live cell imaging, but even the best current live cell imaging technique does not produce image clarity and quality that we need. Fixed cell imaging does not only allow embryos to be optically cleared, but also lets us make probes to any proteins or mRNA of sort of interest. And here I compare two-photon imaging at the top of the best current live cell imaging technique and our current fixed cell embryo work. So, thus, our strategy is to stick with fixed cell imaging and use morphological complexity to classify in a top-down approach the 40,000 cells in each embryo by tissue type. To build an atlas, we need data from thousands of embryos, but we can only label 2 or 3 gene expression patterns in each. We can accurately annotate images by hand, but this is slow. Thus, we need to train sort of machines how to recognize tissues automatically, and this is a big challenge. The way we are addressing this, we are acquiring high-resolution images. We're acquiring two stacks of this and using a best matching overlay of the nuclear positions using translation, rotation, and image scaling to assemble the final images. The raw images are scaled down in XY so that the resolution of the image is isotropic in X, Y, and Z. The images are then automatically oriented along their anterior, posterior, and dorsal ventral directions. And so here, it does run. I show just a movie of one of these assembled image stacks, just to show you the kind of clarity you can see. My pointer. So this is top to bottom, right the way through an animal, 40,000 cells. The ventral nerve cord at the bottom. So, to annotate images, we have built an annotation tool which allows a user to partially annotate certain tissues, and these are completed computationally using methods, image analysis methods on the grayscale images. These annotations are then used to create three-dimensional models of the sort of tissue. And so here I show the hindgut, midgut, the two salivary glands left and right, and the pharyngeal and sort of muscles. And we can do this for a whole set of embryos. This is a movie here just showing some of the dynamics that involved through, this is stage 16, or the biological variability. We just don't quite know what yet because this is making that. So then these models allow us to build tissue-specific segmentation-like analysis to isolate individual nuclei of specific tissues. This is necessary because the shape and packing density of different nuclei differ from tissue to tissue. So using the tissue annotations, we can now train linear support vector machine to recognize specific tissues. The data are split into a training and test set, and each image has approximately 24 million voxels, and each voxel in every image is assigned a unique spatial feature derived from the relative position of the other surrounding nuclei. These features are pyramid gradient or shape context features, which collectively give unique identifiers for each tissue that can be learnt by a support vector machine. The output is a linear classifier that is used on unseen test data to give per voxel estimates of the probability that each voxel is part of any specific tissue. So here I'm showing results of the linear classifier applied to a single test image. The results show as a heat map the probabilities that voxels are part of specific tissues. These three-dimensional results are projected along the dorsal ventral, the left, right, and the anterior, posterior directions here for pharyngeal muscle in the wall and for the salivary glands. And then what we can compare these predictions to are the hand annotations, what we call the gold standard in this image. And so I can just flick back and forth. So this is, again, this is the hand annotation projected there, and here is the sort of prediction here. And if your eyes are good, you can tell that we're missing lots of lots of areas, but we're doing a pretty good job. So the results of this from my vision expert people are the average precision of the classifier, which can be interpreted as the percentage accuracy of detecting nuclei in any annotated region. For example, the pharyngeal wall here, we are able to accurately predict about 330 out of 378 cells that we think are there. And this is impressive given that there are about 47,000 cells in that particular image. It's like trying to find 370 needles in a haystack of 47,000. We're finding 330 of them, so we're doing it at about 87%. So what I've told you here is about using hand annotation to train support vector machines, but of course we also have the gene expression patterns that we're recording along with these data, and this is for the salivary glands here, and left and right. And we are also using these data so for sort of annotation. But in conclusion, we're building an atlas of Drosophila and embryogenesis. It's a digital atlas for studying gene expression, morphology, and embryo dynamics at single-cell resolution. I've outlined our strategy for stage 16, showed some of the progress we've made. Our goal is to create maps of several stages through embryogenesis and to link these via computational analysis using live cell and fixed cell imaging. So I'd like to thank my collaborators, Mark, Jitendra, Pablo, John, and Soila, and the NIH for funding this exciting project.
That's very cool work, David. Thanks very much. Questions for David? I'd like the next speaker to start setting up. David, I had a question on the first part of building the atlas. So obviously this is incredibly high resolution data. For people who are trying to understand gene regulation and would like access to this kind of data, a question is whether this data is too high resolution for any available expression analysis techniques, or is there something you can say with this kind of data that could not have been said with lower resolution?
Even the high resolution for the Barcelona project, for example, we've made an atlas out of thousands of embryos. But because it's just a flat table, these are XYZ sort of coordinates. I think is it 24 megabytes, the whole table for, are we at, not 1,000 genes, but, and so it's relatively publicly and available. You can run it on your laptop. We have visualization tools. You can visualize this embryo, rotate it around. You can show it to biologists.
My question was, if you try to understand gene regulation, regulatory relationships, cis-regulatory code, etc., from this kind of data, is there something you can learn that you wouldn't have learned using existing techniques with lower resolution? That's, are the analysis tools ready for this kind of data?
Well, it's a good question, but what we have here is we have all the spatial and sort of information. Okay. I know I danced around your, but please speak to my sort of collaborator on the biology, Mark Big, and
All right, let's thank David once again.